I think it's a pleasure and an honour to be here. It's a great meeting, and I feel very much amongst friends and people with very much a common interest. Actually, most of what I want to, we've also heard a very good illustration of exactly the sort of benefit that these devices give in clinical practice. As you say, we come from the far end of the world. In our hemisphere, there's not much. There's a whole lot of Pacific. There's quite a bit of Antarctic and a whole lot of Australia, and not much else. And despite, by European standards, relatively low latitudes, where I come from in Christchurch, which is about there, if it works. No, it's not going to work. We're at about 45 degrees, but we have we have an interesting climate. But you can find signs of us everywhere. Okay. I thought I'd spend a couple of minutes talking about some of the things I played with years ago, which ended up, which led to me getting the Navigator and to some extent the Smart Pilot to play with. This was a very early attempt at intravenous anaesthesia in the late 80s. If you follow this recipe, you have the E and the T part of a BET scheme, and it actually produces pretty reasonable levels. The only problem with it, of course, is like all of these stepped infusion schemes, is you can't control the level once you're there. Then probably a decade later, started to say, well, why can't we think about volatiles in the same way that we starting to use intravenous drugs? And we were getting these TCI pumps, which have effect site forward prediction. We've heard the value about that from Dwayne. And what we were thinking is, well, why can't we do exactly the same sort of thing with infusion? So we built a system which did. So at the top, I won't spend a lot of time on this, is the display of our system, which we've now sort of retired, but it shows a forward prediction of both the end tidal and the effect site sevoflurane concentration. In this particular case, you can see that the sevoflurane dial has actually been turned right down to zero and a fresh gas flow rate of just over 300 mils, and you can see what's going to happen over the next 10 minutes. And we did that simply by taking the data that the Datex ADU produced, modelled it, and corrected it for what was being measured. And a very similar looking display is now on the Draeger Perseus. And that led us into work looking at trying to just come up with data which shows to Joe the anaesthetist rather than Joe the plumber the value of taking an effect site approach to anaesthesia delivery. We have a whole range of practice in our department. This is Wayne Morris, who I think is somewhere in the Middle East at the moment. He's very much involved in the World Federation and teaching. Okay, this is our current setup. You've got to ignore the screen off to the far left. That's nothing to do with us. We have navigators in about two-thirds of our theatres, and that's what I'm going to spend the next few minutes talking about. A lot of this has already been answered, really, by the previous two speakers. What is it, what use is it, and what does it show us? Here's the summary. It shows you effect site concentrations and interaction predictions. It's based on the inputs, the data that goes in. It's like, as we heard, any model is only as good as the numbers that go in, rubbish out, and you get these graphical displays of interaction possibilities. Here's my summary slide. I think these interaction devices are a far bigger step than TCI, even though we're talking about manual control. The ways we approach the kinetics of our drugs are overly simplistic. That's because we can't do it in our head, but most people won't, I think, admit to themselves that all of this stuff is far too hard. There may be some people in this room who know this stuff. I can't. I need all the help I can get. The display becomes another part of your monitoring setup. And I think you know, when I started anaesthesia, we thought we knew everything about physiology, and we didn't know much about pharmacology, except we had to learn a whole lot of facts. Now I think it's almost turned around the other way. I mean, you know, things that we were brought up believing in, like the value of the central venous pressure, now we don't know what that means anymore. So here's the Navigator display. They have cleaned it up slightly since the version that Dwayne saw. There's the drug information across the top with the doses. Down the right-hand side are the current effect site values and the total doses given. There's a whole lot of drugs that it accepts either from a manual input or directly from a pump. Atracurium, morphine, and the alpha-2 agonists are the main things missing from that list, which does cause some questions from users. It's got these three domains for the output: sedation, analgesia, and relaxation. It's not really sedation, of course. What it's talking about here is the probability of unconsciousness, and in an anaesthesia context, the area labelled analgesia is the probability of no response to laryngoscopy. That's the specific response that it's calibrated against. So as we've heard, we're getting EC50s and EC95 bands. The effect of the individual drug, in this case a propofol infusion in yellow, heading along the bottom of the band, and you can see the added effect of a remifentanil infusion. Excuse me. We add in the analgesia, and you see two things. You can see the effect of the combined effect. You can also see that the individual drug, and this comes back to the isobologram, which Rick showed us before, that amount of fentanyl, about 5 nanograms, isn't enough to produce to block the response to noxious stimulus by itself. It's barely on the scale, but with the amount of propofol that's there, you can see the synergy quite clearly. And this allows us to give manual target control, not just infusions, it's manual controlled drug delivery, because we've got the forward prediction. And I think, and as Dwayne says, that's one of the really powerful things about these technologies. So the user controls the delivery. You can do the same trick with TCI pumps, of course. It also allows us to deal with drugs that are not in TCI pumps. We heard about sufentanil. We don't have that in New Zealand, but we can go back to infusing relaxants, and it's interesting to remember that a lot of the science around PKPD was actually developed years ago with relaxants, because you can measure a good endpoint. And fentanyl infusions are becoming relatively common in our practice, and it provides a guide. So here's just a fairly standard example of someone giving a propofol infusion and some boluses of fentanyl. The blue line represents the effect site fentanyl, and you can see what's going to happen with that over time. And we're starting to get some idea of what sort of levels patients need at different stages in the procedures. Here's a sort of slightly longer, more complicated case. There's propofol and remifentanil infusions, and you can, in this case, the funny orange jaggedy line is the entropy. They're actually the two values, which is why it's an odd colour, and the scale is flipped. So an entropy orbis, if you were plotting that, of 0 is at the top of the screen. So it moves in the same way as the combined effect. But you get bored after a while with giving all these boluses, and so the logical thing to do is plug in some infusions. In this case, of fentanyl and remifentanil. You can of fentanyl and rocuronium. You can see it took a couple of goes to get the rocuronium infusion up to about the right level, and I must admit I always like to have the relaxant infusion set up so that it's tending to fade off rather than build up. Titrating to effects, the next thing you can do with it. You can see here that the combined effects of both the hypnosis and the response to noxious stimulus have stepped up, and we've done the same with the relaxant. I think in this case we're waited for the surgeon for some time, and then those steps represent the point at which they finally decided to turn up. So you can adjust the effect that you want very nicely to match stimulus. We heard exactly that with Dwayne's study and Joe's herniorrhaphy. People worry, well, certainly a question I frequently get is, well, what about this, that, or the other drug which isn't in the system? I would say that individuals are so variable that it just represents a sort of an offset for that particular individual. And in fact, what we do is what we all do is we use our clinical skill to work out how much the particular patient needs, and the system allows us to stay there. And this quote puts it very nicely. The major strength of effect site controlled TCI, and we could put interaction display systems in there instead, lies not in predicting the resulting hypnotic effect in the individual patient, but rather the ability to maintain the pharmacological condition once a predetermined clinical effect has been reached. And I think that sums this up very nicely. Here's another. This is just an example of what you can do, or if you like, not do. Here we've got about an hour and a half of a relatively complicated case. In fact, nothing much has happened there. There have been a couple of syringe changes along the way, and towards the end, the relaxant syringe thing has been turned down. But that's all the activity that's been needed. You can of course argue whether that's a good thing or a bad thing, but it actually. I will skip over that one. But reducing workload is a good thing. I want to move on to just some specific sort of illustrations of different components of anaesthesia. Here's an IV induction. We've heard from Rick about that. You can see how long the propofol lasts. You can see differing effects. And what it explains very nicely is when the resident is when we don't have such good residents who can smoothly put the laryngeal mask in, they reach for more propofol, but actually it's the wrong drug. Innovational induction is an interesting one. But again, this is demonstrated quite nicely. At this point in time, half the people are asleep. But look where they are on the noxious stimulus response curve. They're hardly starting to. They will respond, and we know that. You have to wait another 2 or 3 minutes to even get up to the 50% unresponsiveness. And if you're going to put a laryngeal mask in, you probably have to wait another sort of 3 or 4 minutes after that as well. So it's all about illustrating effects. We see the same thing with relaxant kinetics, and again, the dosing and variability has changed a bit. This was a fascinating case in a girl with a nasty head injury who we just kept on putting more and more relaxants in, and it took a lot to get it took a lot to produce an effect, but with the Navigator and the bottom part of the screen, I'll skip over it, shows the train of fall response, we got an understanding of what was going on. Analgesics, the graph on the right just shows some work we've done matching intraoperative and post-operative analgesic requirements. You can get a fair idea of how much people are going to need afterwards for what they need from what they've needed during the case. And I often end up using 4 infusions if I'm doing TIVA. The other thing we've seen is that people are more and more taking this information into other domains, and when they don't have the effect site, when they don't have the interaction display, they will use one of the several apps that are around to control their titrate their fentanyl dosing, having learnt how much patients need. Recovery is interesting. The Utah group made the statement that 90% wake within 1 minute of the EC50. We've been testing that in a real-world environment. Now, we use a slightly different endpoint in that we're using the old-fashioned MAC awake rather than the shake and shout endpoint. So our patients wake up slightly later than the system says they would, about 2.5 minutes after they reach the prediction. What the fascinating thing about this, though, is that in a real-world population is how broad that population is, and that's fairly consistent. And I think that's the thing we don't appreciate with our drugs, is how variable the response is. People look at something like the Navigator or the Smart Pilot and they say, what number should I aim for? And you say, no, there's not a number. You find out what number that patient needs. And I think this variability in where patients wake up helps illustrate that. There's more in the poster out the back on this. So here's my summary of what you can expect from Navigator. It really just reinforces what we've said before. The principles can be applied. It's a great learning tool, both in terms of teaching and in other concepts. And it deals with patients. It's got some limitations. Only 3 pumps. The display format we've heard of. The way it collects the inhalational data seems a bit, it's not as good as it could be, shall we say. Some of the individual drug level displays are not as clear as they could be, and there's some drugs missing. And I just want to very quickly go on to our experience. I want to skip ahead, steal some of Jan's thunder, and talk about some of our experience with ISIS and entitled control. Automated systems are good at controlling vapour. This is just an example of the ISIS entitled controller doing its thing. What I want to talk about briefly is what else can we learn from these devices. This is a favourite example. This is the sweep speed slowed down to improve the example. On the left is fresh gas control, 1 litre a minute. On the right is entitled control. The middle plot is the vapour concentration, and in both of these the CO2 absorber has been changed, and the one on the right it needed to be changed, and the one on the left we did it for demonstration purposes. And you can see how long it took. Well, in fact, but after about 25 breaths in the fresh gas example, the entitled concentration is still only about 60% of what it was. The entitled controller gets you there in about 10 breaths. Now, that's, to me, quite a powerful demonstration of how efficient these systems are. You can see the uptake decreasing. Here's a constant entitle, and the inspired concentration is coming down. Here's another example, which I think is a lot of fun. I'm not quite sure what we do with it, but this is off-pump cardiac surgery. So at this point here, the surgeon picks up the heart. The entitled CO2 drops, as you would expect, but so does the inspired concentration, and it drops by a lot. And I think one of the things about this technology, in the same way that the Navigator teaches us a whole lot of things, by watching the way these controllers behave, we can actually learn a lot about kinetics and the way our drugs work. Right. There are always naysayers with any new technology, and this applies to both to all of the things we're talking about today. We know machines are better. People need to learn to give a proper anaesthetic, whatever that is. In a world where TCI is common, do we make our trainees always give propofol by mils per hour? It's useful skill. As I say, watching the controller is often teaches you more than learning a recipe. What if it breaks? Well, of course, you need to be able to cope with that. Expensive new toys actually save money, and I haven't put and prove the quality of the patient experience, but I think Rick would agree with me that we actually are providing better care. Finally, I want to finish with our gas flow data, which some of you have seen. Here's where we were collecting flow data for years from the ADU. This depends which audience, which slide I show this to. Here's some data that we published last year, and here are some published American data for comparison. The two on the right represent a single hospital before and after a specific intervention. For us in our hospital, a half a litre decrease in the mean fresh gas flow represents a saving of about €100,000, and is equivalent to about 100 tonnes of CO2. And these sorts of findings have been shown by others. The other thing, of course, is the decreased number of interventions that you get. Here's a more complete graph of our data, and it's interesting that we had this big jump up when we first introduced the ISIS. I've got my ideas on why that was, but it does mean that when you're comparing the performance of an individual machine, you've got to be careful, because they may actually be optimised for a particular way of functioning and not work particularly well in other ways. But I have to show you our results from just before we came. I went and harvested the data from some of our newer machines. We're now getting an average fresh gas flow rate of about just over 800 mils an hour. That includes inductions. We don't have induction rooms. Part of what we're seeing is that use of end-tidal control is about 85% of when vapour is being used, but we're also seeing some changes in behaviour. So compared to 2009, we're saving about €50,000 per annum, which more than pays for the additional technology. And we've come a long way in the last sort of 15 or 20 years. The machine on the right would have been state of the art about 18 years ago. Thank you very much.
Thank you very much.